Hello students. Yesterday we have started with the chapter photosynthesis and we have already gone through the basics of photosynthesis. Where does actually photosynthesis takes place? Today we will start with the chloroplast pigments. The first pigment which is the most important pigment found in the chloroplast is chlorophyll, and which is green in color. Actually chlorophyll is of seven types, like chlorophyll A, B, C, D, E, bacteriochlorophyll and bacterioviridin. Okay, and these molecules are made up of a squarish tetrapyrrolic ring called the head and a phytol alcohol called the tail. And as I have told you that magnesium atom is present centrally of the tetrapyrrolic ring. Now the four pyrrole rings of the porphyrin head is linked together by the methane, that is the CH group, and is made up of a 4 carbon and 1 nitrogen molecule. Chlorophyll A is actually blue black, while chlorophyll B is green black, and both are soluble in organic solvents. The next type of pigments which are found in you will see chlorophyll B is yellow green, and the next type of pigments which are found in the chloroplast are the carotenoid pigments. Carotenoid pigments are also of two types. The first one is the carotene, which is orange in color and which was actually isolated from the carrot. These absorb light energy to transfer it to chlorophyll A and thus act as accessory pigments. Okay. Also, these protect chlorophyll molecules from photo-oxidation by picking up the nascent O2, that means only the O2 atom, and convert it to harmless molecular stage. The second type of carotenoid pigment is the xanthophylls, which is yellow in color, and it also contains oxygen along with carbon and hydrogen. Lutein is widely distributed xanthophyll and is responsible for yellow color in the autumn foliage. The third type of pigment which is found in the chloroplast are the phycobilins. These pigments are tetrapyrrolic in structure and do not bear the magnesium central atom, and they also don't have the phytol chain, right? Phycocyanin and phycoerythrin together form the phycobilins. Okay, now what are actually pigments? These are the substances that have the ability to absorb light at a specific wavelength, and the various types of pigments which is present in plants can be separated by the process of chromatography, paper chromatography. Okay, now each pigment has different wavelength at which the light is being trapped. The main pigment is chlorophyll A, which actually traps light energy, that is the photons, and the other pigments actually help this chlorophyll A to trap the photons. Okay, now coming to the nature of light. Quantum, we have already gone through that quantum represents the energy contained in the one photon. Okay, now we will come to the photosynthetic unit. Okay, now photosynthetic unit or quantasome, it can be defined as the number of pigment molecules required to effect one single photochemical act and the release of oxygen molecule. Actually it has a photocenter which consists of a dimer of special chlorophyll A molecules, that is P700 and P680. This reaction center actually absorbs the light energy at longer wavelengths. Now the harvesting molecules, that means the molecules which help them to absorb the light energy, they form a protein based complex called the light harvesting complex. And this light harvesting complex consists of two parts. It is in short we call it LHC. Okay, so the light harvesting complex consists of the two parts, that is the antenna and the core molecules. Okay, now the antenna molecules actually absorb the light of various wavelengths, but the shorter they actually absorb light of wavelength which are shorter than the photocenter, that means P7, which are shorter than P700 and P680. On absorption of light energy, these antenna molecules actually get excited, and these excited antenna molecules energy to they transfer this energy to the core molecules by resonance, and when this energy is being transferred, the antenna molecules come back to their original energy state, that is the ground state. Okay, and the energy is being picked up by the core molecules, is supplied to trap to the photocenter. Okay, from here we will come to a Hill reaction, which was discovered by scientist Hill, and it is defined as the production of oxygen without accompanying the CO2 fixation. That means the CO2 is not yet being utilized yet, but O2 is being produced. How? By isolated chloroplast or intact cells illuminated in the presence of suitable electron acceptor like ferricyanide. Okay, so this is the Hill reaction. Now next we'll start with the phases of photosynthesis in my next video. Today up to this. Thank you so much.